Okay, Bismillahirrahmanirrahim. Assalamu alaikum. This is Biotechnology B, and today we are going to talk about biostatistical analysis or statistics in biological sciences. So, handling the biological data in context of statistical analysis is tough in sometimes for a biological scientist because our expertise resides in the laboratory based experiments and some sort of in silico experiments as well in context of bioinformatics. So let's talk about it in this video. Here is an example of bioremediation of water. uh, so this is the pH, this is the BOD, COD, TDS and TSS tests that are further utilized to check the quality of a water sample actually. And this is the dye decolorization parameter, root length, shoot length and seed germination. So first let's understand what this data says. So I have taken it from this research article that was published back in 2023 in the Springer Link journal. They actually did what? They actually collected textile dye effluents. That is, we all know that there are certain textile industries in which the textile is formulated, and after its formulation it is dyed as well. So its dyes include azodye and reactive dyes as well that are considered to be carcinogenic in nature as well. So in this research article they simply collected these dyes from the disposing areas. They collected bacteria from these samples, and then they inoculated these bacteria into the textile dyes that was crude, or that was commercial dyes, that was not the disposed ones, to check either these bacteria are useful to degrade this sort of dye mixture or not. So this, the black color one is shown over here in this picture, is the one that is not treated with the bacteria, and another one that is palish in color, it has been treated with the bacteria. uh, yeah, and they also assessed these bacteria for plastic degradation. Afterwards they collected that particular degraded dye metabolite and they irrigated it into the radish plants and they analyzed their growth. So the data what we just saw in the previous slide, it contained the shoot and the root length and the seed germination percentage. So it was belonging to this particular thing, right? They did further analysis as well, such as the DNA extraction with the bacteria for their 16s RNA identification, the bioinformatics analysis and the enzymes predictions as well. But right now we are going to talk about the chunk from the textile dye degradation and the and their utility for the irrigation of crops. So here it is, this is the data. Now you can understand the pH reduction, the BOD, COD, TDS and other parameters that are mentioned right here. So actually this is the dummy data, I want to mention it right here. It does not contain the exactly same values as that are present in the research article. Okay, so most of the time the life scientists prefer the correlation analysis, and in this research article what I am talking about contains the correlation analysis as well. uh, there are several types of the correlation, such as the Pearson correlation, the Spearman rank correlation and the Candle Tau correlation. Pearson correlation is for the normally distributed data, and the Candle Tau as well, but the Candle Tau is for the ordinal sort of data. However, when we talk about the Spearman rank correlation, it does not depend on the type of the data. That means if the data is normally distributed or if the data is not normally distributed, then we can go ahead for Spearman rank correlation. Correlation tests actually tells us about the association of one variable with other variables. Okay. for this purpose, we have certain softwares such as Strata, SPSS, or RStudio. But RStudio is more preferable because the quality of graphs and because the quality of the pictures we get, because of its user-friendly nature. If you know the R language, then it is super easy for you. So let's have a look how actually R works. So right here I have my script. So first of all, you need to put all the things in a single folder, such as this one, right? So this is the folder where I have my dummy die sample and its corresponding values and everything in the form of a CSV file. You can use an Excel file as well, but right now I'm gonna work with the CSV. This is the file I'm gonna show you right here. Okay, it's open. Perfect. So right now we are in our studio. First we are gonna upload the data on the RStudio. So this is the name. I can name it differently as well. I can name it my data. Sorry, my data. Okay, and this is the sign of vector. We always need to put it when we create a vector. So I am currently creating a vector named as my data, and because we need to make it a data frame, so that is why we are putting that way. And raw names equals to 1, it shows that our data contains first row as the name, so that it will not count the first row as the numerical values, otherwise it is gonna give you an error. So we will press this button, or else we will press control and enter on your keypads. So okay, so right here we have loaded our data in the form of data frame, and it is named as my data. If I write here my underscore data, it comes right here as well. So my data, if I click it right here, so it is showing that the data that is with this name is actually my data. So first we will just upload the libraries what we are gonna utilize right now. But if you don't have these in your library, so first install them through this command line pheatmap, so that it will be installed. I'm not gonna put enter on it because I already have these packages. Okay, so further I'm gonna change the name because I have changed the vector name. I'm not gonna perform Pearson, I'm gonna perform Spearman rank test. So to avoid the errors, first we are gonna read that particular file and mention it to not to read the raw one as a numeric data. So let's do it, and then we are gonna turn it into a data frame, and then we are gonna check it, the data we have just turned into the data frame. So this is the data right here, it has been converted into the data frame. So this is the simple data, this is the simple form of your data, right? Then library the required packages. If you don't have it, then do install them, but I have it, that's why I'm not gonna install them again. You can name it whatever you want, and this is the function for correlation. So I have put it the function of correlation, and in the bracket I have written my data, that is my vector, what I have already turned it into the data frame. Along with that, I am mentioning the method, that is the Pearson method. I can go for the Spearman test, the Pearson test, and whatever I want, but right now I'm gonna work for the Pearson test. So yes, okay, so I have done it. Let me see what I have done. So this is the data. These numeric values, these are the numeric values that show the Pearson correlation of all of these parameters among each other. And as we all know that the correlation lies between 1 and 0. So all of the values are between 1 and 0. Okay, so let's go ahead. P heat map, this is a package to create the heat maps, and I have written this my vector that contains the correlation matrix. So first of all, I have written its name, comma. You can put comma along with a space or along with enter. Moreover, there are certain parameters such as the cluster columns. True means you want them to be there, and if you write false instead of true, it means you don't want it to be there, you want to remove it from your figure. So you are showing the column names, you are showing the row names as well, and you have mentioned the name as the gene expression correlations right here. But if I want to change it as bioremediation of textile effluents, so you can change it under the uh, apostrophe commas, and also you can change the font sizes. Right here I have adjusted as 10 and 5. You can also adjust your cell width and cell height, and these are the colors. I have turned it into light pink and sky blue. You can change it as well, so I can do it like purple. No, light purple would suit more. No, it doesn't even exist. Okay, purple, purple and sky blue. So this is the image I have got from the entire command line. So I can save it, and I can alter its width and height as well. So what if I change it into 1000 width and 1000 height, and I am gonna put it as whatever I want in context of the format, but I'm gonna put it as it is TIFF because it is the most highly resolution containing picture. I'm gonna save it up and we can have a look on it. So this is the picture, this is the scale of the correlation analysis that shows 1 till 0.96. So actually this scale is set up based on the correlation what you got from the entire Pearson or Spearman or whatever you have done the analysis type. So again I would say that the analysis of your data actually depends on what you want to know about your data, right? The data is entirely in a discrete form, so it is very scattered. I'm talking about the raw data. Okay, it is very scattered. So what do you actually want to know about your data? Which parameter you want to highlight, in what way, and which kind of data you have? Either it is a categorical one or either it is a quantitative one. So it actually depends on each of these parameters. Once you got to know the answers of all of these, you can easily analyze your data in the actual way you want. So, this is it for the biological statistics based on correlation on RStudio.